Thank you for that kind introduction. I am Andrew Babiskin, and it is my pleasure to be the moderator for the current session. We will be discussing the regulatory utility and knowledge gaps related to implementing modeling simulation approaches for orally inhaled products, including computational fluid dynamics, coupled with physiological-based pharmacokinetic models. We have the privilege of opening our session with talks from two distinguished experts in this field. The first talk is by Danny Brinkley, who is the Director of Global Inhalation R&D at Teva Pharmaceuticals. He began his career at GlaxoSmithKline and spent 10 years working in respiratory R&D. He then spent 4 years working in an early phase respiratory R&D at Novartis before joining Teva in 2012. He has a passion for understanding the intricacies of IVIVC and how this can be applied to improve the efficiency of respiratory product development. Today, he will share his insights on current limitations in producing a fully mechanistic PBPK model for a highly soluble orally inhaled drug product that exhibits slow lung absorption. The second talk is by Dr. André Jacquas, who is the Chief Technical Officer and Senior Vice President for Research at CFD Research Corporation, where he heads the development of computational pharmacology tools for military and civilian applications. He has been the PI on several projects with DARPA, DoD, FDA, NIH, CDC, and U.S. Pharma in collaboration with academic institutions, and has published over 290 papers as well as several book chapters. Today, he will share his insights on integrated computational fluid dynamics, physiology, pharmacokinetics tools for development and evaluation of orally inhaled drug products.
Thank you for that introduction. In recent years, there has been some impressive progression in IVIVC for respiratory products with mechanistic PBPK models published for drugs such as fluticasone propionate. To date, much of the focus has been on solving the mystery of the mechanisms driving the PK for these polysoluble inhaled drugs, but today I'm going to switch to modelling PK for highly soluble inhaled drugs that exhibit characteristics of slow lung absorption into the systemic circulation. I will present the correlations observed with total lung dose and progress to mechanistic PBPK modelling and the challenges in producing such a model. Before I continue, I must present this disclaimer. And now for the rest of the presentation. Tiotropium Bromide has been selected as a model compound for this study due to the rich body of published information. This table demonstrates that Tiotropium Bromide is a hydrophilic, soluble, cationic drug with low permeability, limited tissue affinity, and a muscarinic receptor dissociation rate ranging from 88 minutes to 55 hours. Another factor to note is the equilibrium dissociation constant for each of the receptors that may determine the extent at which each of the receptors effectively sequester any excess drug. We have observed a PK correlation with lung dose in a 2019 publication by Perry et al. Titled Particle Size and Gastrointestinal Absorption Influence Tiotropium Pharmacokinetics, a pilot bioequivalent study of PUR0200 and Spiriva hand inhaler. The paper assessed the PK of 5 spray-dried tiotropium formulations against Spiriva hand inhaler. The Spiriva hand inhaler arm was dosed both with and without charcoal blockade, while the spray-dried formulations were dosed without charcoal blockade. APSD characterization was performed using a next-generation impactor with an Alberta idealized throat. This graph demonstrates a strong correlation by linear regression between the mean impactor size mass values plotted on the x-axis and the PK Cmax, AUC to half an hour, AUC to 8 hours plotted respectively on the free y-axis. For this graph, we have only used the non-charcoal hand inhaler data. To further investigate this observed lung dose PK correlation for inhaled tiotropium, we developed a mechanistic PBPK model in Preludium using the Spiriva hand inhaler APSD and PK data from the Perry et al. Paper. Preludium predicts the fraction of drug deposited within specific regions of the lung and infers the relative contribution of each lung region to systemic absorption, considering factors such as permeability, dissolution, type of cell transport, and disease state. Deposition modelling includes a lung model which reflects the anatomical complexities of the lung with respect to the various regional bifurcations. APSD data is regressed using a 3-parameter model that considers coarse fraction, MMAD and GSD, and fractional deposition is calculated in the summary lung regions, with ET representing mouth throat, uppercase BB tracheobronchial, lowercase bb bronchiolar, AI alveolar interstitial, and the exhaled fraction EX. In addition, the relative contribution of each lung region to systemic absorption is determined considering factors such as permeability, dissolution, type of cell transport, and disease state. This slide presents a graph comparing observed and simulated PK for the Spiriva Handyhaler non-charcoal arm of the PERI study produced by our first PBPK model. This model overestimated the rate of uptake of inhaled tiotropium bromide to an extent at which AUC to 8 hours was overestimated by 4 to 6 times, indicating that there was a slower or less complete than anticipated absorption from the lung. This suggests that a fraction of the inhaled tiotropium bromide is either absorbed very slowly from the lung or that it is metabolized in the lung. Since the latter is not reported in literature, it can be assumed that the lower than anticipated exposure is due to slow absorption, presumably due to either low permeability, binding to tissue, or to the muscarinic receptors, or a combination of all these processes. Our second simulation attempt, presented here, applied fitted parameters for a putative deep compartment and reduced permeability. This resulted in simulated PK data comparable to those observed. The model is now not fully mechanistic with respect to the absorption process in lung, given that tissue binding and permeability input parameters now are fitted. However, the deposition model is still mechanistic, and model outputs for the systemic PK are thus believed to respond to changes to product performance in a realistic fashion. We will now assess this further in the next slide. On this slide, the two linear regression graphs compare the observed and simulated respective Cmax and AUC to 8 hour values for the Spiriva Handyhaler and PUR0200 spray-dried formulations from the 2019 PERI et al. Paper that have been simulated using our second model. For Cmax, there is a R² correlation of 0.907, with Spiriva Handyhaler observed simulation ratio of 0.8. The PUR0200 sample ratios range from 1.1 to 1.4. There is a stronger R² correlation of 0.933 for AUC 0 to 8 hours. The Spiriva Handyhaler observed simulated ratio of 1.0 indicates that the model accurately estimates AUC to 8 hours for this sample. The PUR0200 sample ratios range from 1.0 to 1.3. Overall, the line of fit for both graphs indicates that the accuracy of the simulated results is weakest for the sample with the highest observed Cmax and AUC 8 hour values. To explore the sensitivity of simulated PK parameters to the regional lung deposition distribution for a fixed lung dose, we used the PBPK model to simulate the Cmax and AUC 0 to 8 hours for different regional lung distributions. The 4 regional lung distributions are represented by these 4 pie charts. The associated simulated Cmax and AUC to 8 hours are represented by the dark and light green bars above. Simulation 1 represents the control, where the regional lung distribution and simulated PK parameters are representative of the simulated PK profile shown in the previous slide. Simulation 2 represents the tracheobronchial and bronchiolar deposited fractions swapped, with no significant impact observed on the PK parameters. Simulation 3 represents 10% of the alveolar interstitial fraction redistributed evenly to the tracheobronchial and bronchiolar regions, and we observed the Cmax and AUC values decreasing by 41% and 26% respectively compared to the control. Simulation 4 represents 20% of the alveolar interstitial fraction redistributed evenly to the tracheobronchial and bronchial regions. And we observed Cmax and AUC values decreasing by 84% and 52% respectively compared to the control. We hypothesized that the observed significance of the IR fraction for Cmax may be due to this region having potential for higher levels of sink conditions, potentially increasing dissolution, the epithelium barrier being leakier, and there appearing to be significantly less or possibly no M3 receptor expression for drug binding compared to the central lung. We note that although the AUC to 8 hours follows this trend to a lesser extent, this is unlikely to be a true reflection of AUC to infinity due to the Tiotropium Bromide absorption kinetics extending only out to 8 hours. A significant proportion of the plasma absorption is known to occur beyond this time point, and therefore we expect all lung regions to be of equal significance for AUC to infinity. In conclusion, developing a PBPK model for highly soluble orally inhaled products that exhibit a slow lung absorption component presents a unique challenge. The slow lung absorption component meant that only a partial mechanistic model was possible with fitted parameters required for a putative deep tissue compartment. This model produced a simulated PK profile consistent with the observed profile for SBIA hand inhaler, with simulated observed ratios ranging from 1.0 to 1.2 for Cmax and AUC to 8 hours respectively. The model indicates that the alveolar interstitial region has a significant influence on PKC max. Further research on the mechanisms determining this slow absorption component will be useful when creating a fully mechanistic model for orally inhaled products such as Tiotropium Bromide. This may help to validate the accuracy of the calculated regional lung deposition profiles. I would like to say a big thank you to all of those who have contributed to this presentation, and they are acknowledged here. All literature references from this presentation are listed here. That concludes my presentation. Thank you so much for listening to me. I'll be back later for the panel discussion.
Thank you for that introduction. Today, I will introduce you to the use of computational fluid dynamics technology to aid in the development of orally inhaled drug products. I will briefly describe the basics of CFD lung models, and I will show how the CFD model can be linked to the whole body PBPK, and briefly discuss the main components of the simulation pipeline identified in this panel. I will compare CFD-PBPK model predictions with clinical pharmacokinetic data for 2 representative corticosteroid drugs, and I will close my talk with few recommendations. CFD software tools solve fluid momentum and continuity equations along with transport equations for the dispersed phase, for example, for inhaled aerosols. Generation of 3D computational mesh within human airways for CFD simulation is not trivial. 2 example meshes are shown on this panel. Much more challenging is the solution of the dispersed phase particle dynamics within airways and particle deposition on airway walls. There are 2 methods for this modeling. In Euler-Lagrange method, each particle is individually traced along its trajectory. In the Euler-Euler method, a particle mass transport equation is solved for each particle size. Both have advantages and limitations in accuracy and computational speed, which I will discuss later. In 2 examples shown at the bottom, simulated using COBE tools, dark dots represent the EL and colors the EE results. As can be seen, both methods show similar results. CFD can be used at various stages in the development of orally inhaled drug products. Simulation of inhaler device can provide particle jet dynamic parameters such as particle size distribution, velocities, and amount of API released. It can account for effects of drug product formulation, device design, and inhalation maneuver. CFD tools can also help in modeling conventional in vitro dissolution and permeation devices, as well as transport across airway barriers in novel lung-on-chip type devices. In my presentation, I will focus on three highlighted items: CFD modeling of in vitro and in vivo aerosol delivery, airway transport and deposition, airway barrier model, and coupled airway barrier and whole body PBPK. Note the difference in timescales required for each of the above simulations, marked in blue on this panel. It would be great if CFD model validated on in vitro devices coupled to PBPK could predict in vivo lung tissue concentrations of the inhaled drugs as well as the systemic PK. I think that a first principle-based CFD model should be able to predict drug product performance in various in vitro devices with minimal parameter calibration. Some of such devices already exist, shown on the left, and they are established as commercial products. Simulation of in vivo inhaled product pharmacology, shown on the right, involves two sequential steps. One, short duration aerosol transport in the airway lumen and deposition, followed by longer duration airway barrier transport and absorption simulations coupled to the PBPK. Our vision is that if the same model is used for in vitro and in vivo simulations, it may enable predictive in vivo capabilities. The first step in modeling human airway, drug product inhalation and deposition, is the generation of the whole lung anatomical geometry. As you may know, there is huge anatomical and physiological variability among human lungs. To address this challenge, we have developed a protocol to generate human airway and lung geometries using anthropometric and physiological data. Using principal component analysis, or PCA, trained on thoracic clinical imaging data, we can create personalized and population lung geometry models. For CFD simulation, we can generate computational mesh of the airway, lumen, and the barrier wall, which is coupled to the whole body PBPK. We have used both high-fidelity 3D models and reduced order quasi-3D or Q3D airway models. It is not feasible to generate a 3D mesh in the complete lung airways. To address this challenge, we use two meshing approaches. A 3D model of the upper airways is connected to quasi-3D or Q3D models for distal airways. Another is to use a full Q3D model for the whole airways. In both methods, the alveolar sections are represented as termination trumpets. The models reproduce whole lung volume and airway surface area of an average adult male. As mentioned before, the aerosol dynamics and deposition can be simulated using EL and EE models. Here, we identify their advantages and limitations. Panel on the right shows quite good agreement in modeling airflow in upper airways using 3D and Q3D models. Just described particle deposition model provides initial conditions for the airway barrier model. In our Q3D airway barrier model, the wall at each axial location is subdivided into several layers where we solve undissolved and dissolved drug mass balance equations. A mucociliary convective clearance is simulated in the mucosal gel sol layers. The drug cleared from the lung is passed to the GIT, gastrointestinal tract, of the PBPK model for further absorption. The dissolved drug in the lung is transported across airway wall barrier layers and is absorbed into systemic and pulmonary circulations. The absorbed drug flux is used as input for the PBPK model. The systemic drug can be also reabsorbed from the blood back into the airway barrier. Combined CFD-PBPK model has been validated on selected corticosteroids and is being tested on bronchodilators. Here we present comparison between CFD-PBPK predictions and clinical data for two representative inhaled corticosteroids, highly soluble budesonide and practically insoluble fluticasone propionate. The top figures show the blood concentration of these two drugs after inhalation of 1 milligram dose. These graphs show our simulations in red, discrete data points from several clinical trials, and an average of all clinical data, a black curve. Thing to note is the rapidness of Cmax in budesonide, which is within 10 to 15 minutes, primarily due to the higher solubility, whereas it is almost 70 minutes or so for the fluticasone due to its low solubility. Though solubility is not the only factor here to determine Cmax, the other parameters such as log P, fraction unbound, blood-to-plasma ratio, and gut bioavailability all play the role. In general, very good agreement has been achieved for both drugs. Note that for systemic PK, we have clinical data available, whereas no such experimental data is available for pulmonary tissue concentrations. The lower two graphs are showing the lung tissue concentration in three different lung regions: tracheobronchial region in green, respiratory region in blue, and the terminal alveoli in purple. The overall average lung tissue concentration is shown in red. Similar to systemic drug concentration, what we see here is that once fluticasone is absorbed, it stays in the lung for much longer time compared to budesonide. The model also allows parametric sensitivity studies evaluating effects of log P, blood-to-plasma ratio, and a few. As shown in this presentation, CFD models can support the development of orally inhaled drugs through better understanding of in vitro processes and in vivo pharmacology. More rigorous model validation protocols on in vitro devices are needed for various aspects of pulmonary delivery, deposition, dissolution, permeability, and retention. Predictive capabilities of such model with minimal calibration still needs to be demonstrated. Combined CFD-PBPK can simulate regional lung distribution within the lung barrier as well as in the rest of the body. Further validation of CFD-PBPK models is needed on clinical data for approved and innovative drugs using refined data collection protocols such as pulmonary imaging, plasma data, post-administration, biomarkers, and others. Thank you for your attention.
Well, good afternoon, everyone. My name is Andrew Babiskin. I'm the moderator for the panel discussion for subsession 1A of breakout session number 1 for model integrated evidence for generic drug development. Before we move into the panel discussion, just a quick update to agenda. This panel will actually be running to 2:25 p.m. instead of 2:20 p.m., so additional 5 minutes there. But we will still be starting the next breakout subsession 1B at 2:30 p.m. So the only change is that this one is being extended to 2:25 p.m. So I want to thank the presenters, Danny and Ajay, for their presentations. Before we start the panel, I want to introduce the 3 additional panelists that we'll have with us this afternoon for this subsession. First is Dr. Andrew Cooper, who joined Mylan Global Respiratory Group, now within Viatris, as Head of Analytical and Material Sciences in 2012, based in Sandwich, UK, and responsible for CMC strategies across Viatris' global portfolio of innovative and generic respiratory products. His current interests include understanding the relevance of in vitro tests for in vivo product performance and bioequivalence strategies. Our next panelist is Dr. Gunther Haukas. He was a professor of pharmaceutics at the University of Florida, where he first joined in 1987 as an associate professor. His research is interested in evaluating inhalation drugs through in vitro and pharmacokinetic dynamic approaches. He collaborates with regulatory authorities to improve methodology for drug approval of generic inhalation drugs. Dr. Haukas is a fellow of AAPS and ACCP. Our last panelist is Dr. Ross Rulanga, who joined FDA in 2015 as a ORISE fellow. He is currently a chemical engineer at the Division of Quantitative Methods and Modeling at the Office of Research and Standards, Office of Generic Drugs. His research interests include computational fluid dynamics, modeling of orally inhaled nasal, ophthalmic, and dermal drug products to answer questions pertaining to bioequivalence. For the audience today, please, as we go through the panel, please include any questions or comments that you have that you think is relevant to the discussion. With over 250 people here and only a limited amount of time for the panel, we'll try what we can do to see whether or not what you put is relevant and add it to the conversation. If we're not able to incorporate it, we would certainly look at this after today's workshop and incorporate it into our thinking. Before we get into some more specific discussion topics, I would like to open the floor first to the panel if they had any questions for the speakers for the presentations they just delivered. This could just be a clarification or a request upon any scientific specific item presented on.
I had a question for Danny. It was a wonderful presentation. If I understood it correctly, the mechanism for the long retention in the lung is not known. And you, I believe, scaled it also with respect to the regional deposition, that there was a difference in permeability with the highest permeability in and then lower permeability in the central. Is this a general feature that you can use? Because if we want to use a to make regulatory decisions, one of the questions is differences in regional deposition. And I believe it's relatively straightforward for corticosteroids, where we pretty much know what factors are important for the pulmonary fate. There are a lot of water-soluble drugs, but the retention, I believe, is not known. What should we do with those kind of drugs where we just don't know the mechanism of longer retention in the lung? And how can we use PBPK models to then make regulatory decisions?
So, first of all, thank you for your feedback. I'm afraid you were breaking up a little bit from my end, so I didn't capture everything with your question, but I'll certainly answer what I heard to the best of my ability. So I think in terms of the application for these models, for these types of compounds, I think we need to consider what are we trying to achieve. And there's probably two purposes that I think in terms of my field of work. The first is predicting PK outcomes without having to run multiple PK studies, and understanding how formulation and process parameters can impact that for these type of compounds. And the second is the relevance of these models in terms of understanding local deposition. So with regards to the latter point, I'm afraid one of my slides ended up on the cutting room floor to meet the 10-minute time limits. But if you wanted more information on the deposition model itself, I recommend paper that was published last year by Bachman and Olson. It was titled Pulmonary Drug Dissolution, Regional Retention and Systemic Absorption, Understanding the Interactions through Mechanistic Modelling, and the authors developed a PBPK model for FP in Preludium using comparable methods to what I presented today for tiotropium. In terms of just understanding the mechanisms for driving PK for tiotropium, I think there's some nice papers that are already published using mechanistic style modeling and breaking down the PK into 3 phases. Cmax representing the fast absorbed portion, and then there's a small contribution of the systemic available drug, and then there's that slowly absorbed fraction that contributes to the slowly declining plasma concentrations, correlating to the slow dissociation from the receptors that we are finding very difficult to model. So yeah, I hope that answers your question, but as I say, I didn't hear everything that you asked, I'm afraid.
Thank you.
So I forgot to mention that when speaking, please first introduce yourself. Just identify, this is Andrew, just to help out. Before you move on to see if anyone else has any questions for presenters, anyone want to follow up on this specific question? If not, does anyone else have a question for the presenters?
That's a really good question. That's got me thinking. I'm not sure. I'm not sure, to be honest. I think when it's come to our modelling and considering, especially with tiotropium and the third long phase that we're seeing, and the potential association with the receptors, and correlating that to what we know about regional deposition from the model, we may be able to get some information in terms of optimizing the formulation. But at the moment, from what I've seen from the model, we don't have, or we haven't seen that type of specificity to be able to determine that to that extent at the moment.
Any other follow-up or any other questions?
Yeah, so I have a question for Andre about CFD modeling and its use in BE. So do you think that CFD-based models currently have the discriminating ability to predict exposure differences between OIP test and reference products of the same active ingredient, which would be relevant to development for BE for that product? And if not, what further development of these kind of models and their inputs do you think is required?
This is Ross. I'd like as well. I think that you have to possibly separate between two different scenarios, where in one scenario you're trying to predict from the ground up, like purely mechanistically. So if you have a meter dose inhaler, you're trying to predict the atomization of the spray all the way through deposition. Or if you have a dry powder inhaler, you're trying to predict the deagglomeration of the powder and then deposition. Or you can have another scenario where you're predicting based on experimental measurements. Say you've got your plume angle or your particle size distribution, and then you're using those as inputs. So that's sort of a less stringent case. I think in that case, as long as your experimental measurements are good, I think it's possible to discriminate as far as deposition between the two products. But if you're talking about the more from the ground up mechanistic, I don't think the models are there at this time.
Does anyone else want to add to this before we move on to our first real discussion topic? All right, let's move along. So today we heard from Danny a specific presentation related to the challenges developing a model with a product which has high solubility and slow drug absorption. Tiotropium was the example given there. Given that the purpose of the workshop today is to discover where are the gaps and challenges with employing these models and what research we can undertake to further see these models being used for regulatory decision making. Let's expand this to other type of products. We can go one by one, but the first one we said would take the case of high solubility and rapid uptake in the lung. Does anyone have any additional considerations they would like to add to that case?
So I think if you're talking about bioequivalence in that scenario, you may be able to consider regional deposition predictions standing alone as evidence of bioequivalence. If your dissolution and uptake are rapid enough, then you could consider that maybe the regional absorption and the regional deposition are nearly equivalent.
This is Günther. Ross, I had a question. So some of the in vitro tests are trying to address this question of regional deposition. Could one use computational fluid dynamics to maybe calibrate those in vitro systems? Asking the question, how much different could be cascade impactor profiles be? And we still have a very, very high likelihood of pulmonary bioequivalence. How much different can an MMAP be to be still on the safe side? And I see computational fluid dynamics, at least theoretically, one possibility really would be to calibrate those in vitro systems that we already have.
Yeah, thank you for that. I think that's a good point. I think it's certainly possible to do that. Maybe you could just take the measurements you've got and perform predictions just to explore the maybe so-called safe space around that. It's also even possible to predict the particle deposition in an actual cascade impactor with CFD too, so it might be useful to use a computational technique that way too. But yeah, I think it's a good idea. Yeah, I think those models are really interesting. And I've seen some work in literature developing in vitro tracheal models that extend throughout the entire lung. So I think that those models are pretty are premature at this time, but it seems that there's research going in that direction.
Okay, any other comment? All right, for the sake of time, we just open the floor for the rest of the scenarios, which we also have now the opposite of the last, what we first opened this up for, which is low solubility and slow drug absorption. But we could just, in general, think about low solubility drugs, even those that have rapid intake as well. And if there's also any considerations that should be in play when we're considering the aerosols that are being generated through meter dose inhalers or those through dry powder inhalers.
This is Andrew Cooper here. Having broadened the question to consider all the scenarios at once, maybe I'll make a general point, which I think is that in my view, it's always good to try and model. Not least because I think when you start dividing drugs up into these categories, you are to some extent making assumptions about what you think you know about the way they behave. And so, and I think Danny's example shows this to some degree, that actually by modeling, you actually get to, challenge those assumptions. And it's complicated because you're not just challenging the assumptions in your model, but you're challenging the relevance of the in vitro data you're putting into the model. So you have to kind of interpret the outcomes more than one way. But I think in my view, if you start with the assumptions about what's driving the biopharmaceutics, and then you try and model that, and then you see what that model is telling you, how predictive it is, where the errors are, then you really start to challenge your understanding of what ultimately drives bioequivalence, which is ultimately, from a generic drug perspective, what we're trying to do.
All right, well, we'll move on from this topic because we only have about 3 minutes left. Let's move to the next topic, which is kind of down the road or downstream. Once we actually develop some models that we think are sufficiently validated, is there any thoughts or opinions on what you think the modeling outcomes that should be used to demonstrate equivalence, whether or not it's certain regional lung deposition measures, total lung deposition, or any thoughts about even statistical testing that should be employed while using the models?
Günther, again, I think it boils down again to those 3 questions that need to be answered. Is the dose equivalent that is being deposited in the lung? Is the retention time about equivalent? And is the regional deposition equivalent? And I think modeling can help trying to find parameters that might be able to answer those 3 questions. I believe with respect to dose and pulmonary residence time, even without models, probably pharmacokinetics can answer those questions by just looking at AUC and potentially Cmax. For me personally, the challenge is to answer the question, is the regional deposition equivalent? And depending on what kind of drugs you have, you probably could either go one way and say, I use my models to validate maybe very, very common parameters, Cmax and AUC, as parameters to answer this question of whether the regional deposition is equivalent. And for some of the drugs, definitely that will work. And I believe also Andrew has shown that for lipophilic corticosteroids, Cmax and AUC can answer the question of regional deposition, at least from my experience. The challenge I see for me personally are more the water-soluble drugs like a tiotropium, where we maybe not fully understand the mechanism of retention and how that relates to regional deposition and regional deposition differences. And so that might be one challenge, and which still tells us that we definitely need to know a little bit more about the mechanism of what happens with those kind of drugs, the salmeterols and the tiotropiums, and how is that related to differences in regional deposition. And I think if we make progress there, then PBPK modeling definitely will also help answering those the issues with those kind of drugs.
This is Ross. Go ahead, Andre.
All right, Ross, can you make a quick final comment?
Yeah, this is Ross. Yeah, thanks for the good points, Gunther and Andre. I think like I said before, your ideal scenario is that if you have high solubility, high permeability, just rely on regional deposition alone. But if you don't, then you get into these more complicated situations where you need to understand what's happening in the lung. And the others were saying PBPK and PBPK is useful for trying to better understand these things. So I think that from that modeling, you can potentially identify in vitro studies such as dissolution studies that might be useful for supporting the case of bioequivalence.
Thank you to everyone on the panel for participating today. We'll have to move on to the next subsession, 1B, on modeling for LAIs, which is still going to start at 2:30 p.m. promptly.